Welcome to the Skeptic Zone, the podcast from Australia for science and reason.
Hello and welcome to the Skeptic Zone, episode number 515 for the 2nd of September 2018. Richard Saunders here with you from Sydney, Australia. A little bit cold for spring. And I know the rest of the world's saying, hang on a minute, the seasons haven't changed yet. But for some reason here in Australia, we regard the first day of September as the first day of spring, even if the real first day of spring isn't until a bit later. Go figure that one out. Quite frankly, I wouldn't mind if the first day of spring was delayed until next year, but that's just me. Coming up on this week's episode of the Skeptic Zone, we chat to Associate Adjunct, hang on, is that Adjunct Professor? Professor Adjunct, we chat to Paul Willis, a long-time friend of the Skeptic Zone, dating way back to the early days. Paul used to work at RI-Oz as the director there, and that's when we were bringing you A Week in Science for many years. Paul's going to be talking to us about a recent survey that came out that shows a decline in the belief of, or the belief in creationism amongst university students, which must be a good thing, and his take on the secular outlook. A piece of his mind with Paul Willis coming up at the top of the show. Following that, it's our new reporter, Michelle Franklin from Darwin in the Northern Territory, with her segment, Life is Wild. And Michelle's segments are going to be focusing on wildlife and animal issues. Very interesting. This week, to kick off, she's going to be looking at, are cats in danger of being eaten by snakes? Yep, that's an interesting one. The story of Maggie the Moggie. Then to round off the show, the last part of our Speed Meet the Scientists. That was a wonderful night here in Sydney a couple of weeks ago during Science Week. This week we're going to speed meet a marine archaeologist, an evolutionary biologist, a behavioural neuroscientist, a linguist, and an astronomer. Well, that's enough for me. I'm going to run downstairs, try out some sweet potato pasta, eat that very quickly, come back and bring you the rest of the Skeptic Zone. Of his mind with Professor Paul Willis. And joining me now all the way from Adelaide from Media Engagement Services is none other than Professor Paul Willis. Hello, Paul.
Oh, thank you very much for the promotion. It's actually only an adjunct associate professor, but I'll gladly take the professorship. Thank you very much.
Well, not many people go to the bother of saying all the other things. It's normally just professor, isn't it?
Technically, no. I am only an adjunct associate, but yeah, there you go.
You got it in. Nice to catch up with you, Paul. Long-time listeners to the Skeptic Zone will well remember the contributions you've made to the show over the years, especially with A Week in Science some years ago when you were at RI-Oz. Good times. You've moved on since then. Tell us a little bit about this media engagement.
Look, basically, our main proposition is we can show researchers how to communicate their research using social media. Basically, they can take control of their message, and by doing so, they can actually increase the impact of their research. So we've worked out how to string together various social media platforms so that you create a network that reaches out specifically to the people who are actually interested in your research. I think the basic proposition is, a lot of people go into research communication and they think, I want to tell the world about my research. And it's like, why bother? Why bother going on television, talking to 1 million people who couldn't really give a toss about what you're doing? a much better value proposition is to set up a dedicated audience. It might only be a couple of dozen people, but if those people are actually going to take your research and do something with it, that's the audience you need to be talking to. So that's my basic pitch.
Yeah, very interesting. And just before we get on with the rest of the chat, how can people look at that information online?
mediaengagementservices.com.au. It's all there on our website. We can produce seminars or workshops, video tutorials, any way to get this kind of information across to researchers.
And folks, of course, Paul Willis runs on the board with long experience in the media and on TV, and as many Australians will remember. But right now, Paul, I'm very interested in a story that I think you brought to my attention via a tweet or a Facebook a couple of weeks ago. And it seems fewer Australian university students than ever believe in creationism. And this is sort of research done by our mutual friend, Professor Mike Archer.
Yeah, who was actually my former supervisor. And in fact, in the very year that I started with him as a research student, 1986, he started a regular annual survey of first-year students asking which of four positions they actually hold true. So these are students at the beginning of their university career in science, in biological sciences. And the four options are evolution without any kind of supernatural interference. The second is that evolution has occurred, but there has been a god or some other supernatural influence over the whole process. Then there's adherence to creationism. And the fourth category is don't really know. And he's conducted that now for 32 years? So he's got a really interesting set of data now, which shows some very intriguing trend lines. Basically, creationism amongst first-year biology students at the University of New South Wales has dropped from about 12% down to just a few percent. So there is a significant decline in first-year students who are committed creationists. But the most interesting drop is a very large drop in the number of theistic evolutionists. So those are the ones who tick the box saying evolution has occurred, but it's been guided by God. And commensurate with the drop in theistic evolution has been the rise of non-theistic evolution, of straight, no God-involved scientific evolution. The graphs that are generated, and I can recommend that the listeners go and chase up these reports on this particular study, because the graphs generated, there's a clear X with the decline of theistic evolution and the rise of plain evolutionary biology.
It's interesting because, I mean, ever since I've known you, Paul, that's been one of the major issues you've certainly been concerned with. And if I hark back to the origins of the Australian skeptics, apart from spoon bending in UFOs and all the good stuff from the 1970s, one of the huge issues that the Australian skeptics tackled in the early days was creationism. And these days, it hardly registers on the radar at all. Is that what your finding is as well?
Yeah, my introduction to skepticism was through my work with creationism or against creationism. I was arguing in primary school against teachers who were putting forward the creationist message. And yes, there does seem to be a drop-off in Australia of the creationist message. It doesn't seem to be as widespread or as pervasive as it was even 10 years ago when we had the whole intelligent design debacle.
Yes.
Which I reported on that for Catalyst. And, we went out to a Christian school in the hill suburbs of Sydney, and they were presenting intelligent design in the classroom as, look, this is a possible alternative, makes you think, doesn't it? No, it only makes you think if you don't know what the subject is. It was adequately described as the lipstick on the pig of creationism. And, it looks like from the data that's come in that creationist influence has dropped off. But more importantly, the whole abandoning of theistic evolution, that's really well reflected in the census data, which shows the rise of secularism, of people choosing no religion as their first option on the census.
Yes.
So this rise in not simply the rejection of God, but just moving away from the whole religious question. I think that's what we're actually seeing reflected in this data of first-year students.
Yes, let's pick up on that subject in a moment, but before we do that, I wonder, Paul, if you remember about 10, 11 years ago, you and I and a merry band of skeptics set off and we attended a creationist lecture, complete with a scale model of Noah's Ark here in Sydney.
Look, I've been along to so many of them. They're all as appalling as each other. As with virtually any subject that skepticism tackles, I maintain that we're never going to win the battle. You're never going to convince the other side that they're wrong. But if you don't get in there and battle, they will win, and we can't afford that. The object isn't a skeptical challenge to any supernatural proposition. The object isn't to convince the other person that they're wrong. As I said, you're never going to do that. They're committed. But because of the online environment, there is a large audience out there who are watching, and they're the ones whose minds you can change. They're the ones who are looking for the most reasonable and passionate argument. So, my tip on arguing against creationism or anything that you want to argue online, there's 3 basic rules. Take the high moral ground, always be polite, and always bring it back to the evidence. If you take those 3, then the audience that's watching that discussion are more likely to accept what you're going to say rather than someone who's probably going to start ranting and name-calling and obfuscating.
Now let's look at this whole area of secularism. And it just occurred to me that even though we can't convince the hardcore believers on the other side, there are certainly less of them. Do you think that's partly because simply, and to put it simply, they're dying out?
Yeah. I think that there's also, because now society allows more debate and discussion and questioning of religious propositions, each successive generation is doing exactly that. They're actually questioning religious propositions, and that they simply don't measure up to the expectations that people require for a code of living. Now, you throw into that the abysmal track record of some of the churches when it comes to things like pedophilia in the ranks and the whole same-sex marriage debate. That it's incongruous for many people to think that these are organizations that are promising to give you a moral code, and yet they're behaving so immorally. And so all of these factors, I think, come together to have people abandoning faith and belief as a process to knowledge.
Yes, it's interesting. So, and of course, this has led to, in part, I guess, to the rise of what we might call the broader secular outlook.
Yeah, and, you can say maybe it's the rise of atheism, but I think it actually goes beyond that because, I've been surfing along on the whole rise of atheism over the last decade or more. But I've never really felt comfortable there because essentially, as a proposition, it's not stating what you accept. It's stating what you reject. Mm-hmm. And, we don't define ourselves as, well, I'm not a stamp collector. I mean, what does that tell you about me? You don't expect a Catholic to say, I'm not a Protestant. That doesn't really tell you what they are. And so by taking a rejection statement as your position statement for what you accept in the world, that doesn't make sense to me. But also, there is an underlying thing that really it's not that I reject God, it's that I reject belief as a knowledge system.
Right.
And if you reject belief as a knowledge system, then you can't accept a God, because a God only exists as a belief proposition. You cannot provide evidence that there is a God, and therefore it's a belief statement. So I think that more and more people are coming around to this. It's not a rejection of God. It's a rejection of belief. And that's why I've switched from identifying as an atheist to identifying as a secular humanist. You know, I've now actually become an ambassador for the National Secular Lobby. And the basic proposition is, look, let's have a truly secular society where we have the religious freedom for you to believe and accept whatever you want. I won't challenge that, but you must play by the rules. And there are 3 simple rules for a secular society. You must not do any harm to yourself, to your parishioners, or to anybody else through your religious beliefs or practices. You must not inflict your beliefs on anybody else across society. And you must act within the laws of that society. If you stick within those 3 rules, I don't give a damn what you believe. You know, you can go away and believe what you like, but you must maintain within the bounds of those 3 rules.
That's interesting, and I couldn't help, and I wonder if listeners felt the same way. I couldn't help thinking of Isaac Asimov's robotic rules when you were reading those out.
Well, maybe with the rise of artificial intelligence, maybe that's the way that we do need to phrase the laws of secularism.
Maybe we do. Oh, so you're an ambassador for the National Secular Lobby, and what else does that entail?
Well, let's see. Among our ranks are Chris Schott, he's another ambassador, Jane Caro, Philip Adams, and basically we've been set up to provide an alternative to groups like the Australian Christian Lobby. So in the political arena, there is a secular voice that can be heard above all of the religious voices that are injected in there through various other lobby groups.
That's interesting, because that's been a particular theme, especially, I think, here in New South Wales for a long time, with a hardcore Christian group in the New South Wales Parliament. Yeah,
look, I think when you look across every state and indeed at the federal level, particularly at the federal level, look at this debacle that's just occurred with the leadership of the nation.
Yeah.
And a large chunk of that can be sheeted home to the Conservatives, who are basically, most of them are extremely religious people, and their conservative agenda is closely allied to their religious beliefs. And so how can we have a different conversation, one that is not hijacked by religious beliefs on the political stage, one that gives a secular voice so that all beliefs and non-belief systems have a right to be represented with equal merit on the political stage.
And how can people find out more about the National Secular Lobby?
Oh, how do you find out about anything these days? You Google it. Just Google the bloody thing.
I have to ask, Paul, you know.
Yeah. Look, the National Secular Lobby, we've got a Facebook page, website, Twitter account, Instagram, you name it, all the bells and whistles. just plug National Secular Lobby into your favourite browser, and you will find more than you need to know about us.
Well, Adjunct Associate Professor Paul Willis, I keep promising or threatening to come down to Adelaide to visit you. I'm sure I'll do it one day. But for now, thank you for appearing on the Skeptic Zone.
Always a pleasure, and there's a cellar full of red wine here waiting for you.
Calling all Skeptic Zone listeners. Do you live in or near Glasgow, or are you planning to visit sometime? Then you're in luck, because the Glasgow Skeptics have got your Monday nights sorted. We're committed to filling up every available Monday night with talks on science and skepticism. Past speakers include Eugenie Scott, Jerry Coyne, Michael Marshall, Nate Phelps, Tom and Cecil from Cognitive Dissonance, P.Z. Myers, Richard Wiseman, A.C. Grayling, Noah Heath and Eli from The Scathing Atheist, Simon Singh, Rebecca Watson, and a multitude of local academics and skeptics. There's literally nothing better you can do on a Monday night in Glasgow that doesn't involve taking your clothes off. So come join us. We've also got a vibrant online community. You can find us on Facebook, Twitter, and get involved with the discussion. Glasgow Skeptics. Self-help for your brain.
Life is wild. With Michelle Franklin.
Hello from Darwin, in the top end of the Northern Territory. This is Michelle Franklin reporting in with questions and maybe even some answers about our environment and the creatures that we share it with. So, a friend of a friend asked me recently whether her pet cat was in danger of being eaten by the local wildlife or vice versa. Maggie is a Maine Coon moggie, and she recently moved here from South Africa. She's been through quarantine and she's moved into a suburban house in Brisbane, and all seems to be going pretty well so far. Except that one day recently, Maggie's owners discovered that they have a new housemate, a very large and non-venomous coastal carpet python. I'm going to call her Sally the Snake. Now, of course, Sally's owner is concerned for her pet's welfare. She has been thinking for a while now that she might build an outdoor enclosure so that Maggie can enjoy the sunshine without any unwanted wildlife-related incidents. And the recent addition of Sally to the household has raised a couple of questions that I'd like to explore here. Question number one being, is Maggie in danger? And the short answer is yes, carpet snakes do eat cats. But how often does this happen, and how much danger is Maggie actually in? Now, I'm not a cat person, so I had to Google Maine Coon, and it turns out that they're a pretty big breed of cat. But the thing is, carpet snakes can get pretty big too, and reports are that Sally is a pretty big girl. So, according to a 2001 study published in Wildlife Research that looked at the stomach contents of 258 carpet snakes which were removed from houses in Brisbane and Ipswich, only 2 of those snakes actually had recently eaten a cat. The rest of the prey items listed were various birds, mammals and reptiles, including 17 rats, 8 mice, 4 chickens, 8 budgies, 12 lovebirds, a guinea pig and a fruit bat. And the majority of the prey appeared to be birds. So this in general seems to tell us that carpet snakes are far more likely to eat the local wildlife or the pest rats and mice rather than the pet cats. But there's one small detail that makes a big difference here. The size of the snake was correlated really strongly with the size of the prey item. So the smaller snakes were eating mostly mice and rats and parrots, while the really big snakes were eating the possums and chickens and cats. So not good news for Maggie the moggie. And I think it's probably safe to say that Sally is in danger of being eaten. Question number 2. Is Sally actually a benefit to have around? And there's a couple of aspects to this question. Generally, it's well accepted that having a carpet snake on your roof is good for rodent control. They eat the rats and sometimes possums and things like that. And this is supported by the 2001 stomach contents research that showed a large number of rodents being eaten by carpet snakes. But what about other snakes? There's lots of venomous snakes that share our suburbs and sometimes our homes, and one of the common arguments for keeping harmless snakes around is that they displace the venomous snakes and make our environment safer for us. So, is Sally the snake going to protect her new housemates from her less cuddly cousins? And is the risk of Sally the snake eating Maggie the moggie offset by her presence reducing the risk of Maggie encountering, say, Barry the brown snake? Now, I've heard this claim before. It seems fairly reasonable to think that snakes might be afraid of each other. But when I went searching for evidence to back this up, I couldn't find any. There were studies that listed different types of benefits from having carpet snakes around, but none that I could find said anything about them deterring venomous snakes. So I turned to an expert. Dr. Ruchira Somawira is a herpetologist who specialises in behavioural and evolutionary ecology of reptiles. He said that he doesn't know of any scientific evidence supporting the idea that carpet snakes keep venomous snakes away. And in fact, for many of the large venomous snakes, pythons are actually a source of food. And since pythons generally don't eat other snakes, there really isn't any reason for them to be worried about a carpet snake in the area. Now, absence of evidence is not necessarily evidence of absence, but I feel pretty confident that if this was a real thing, someone would have found evidence and written about it. So I'm going to assume that in this case, it's an urban myth. And while Maggie the moggie is in danger of being eaten by Sally the snake, it's probably reasonable to assume that Barry the brown snake is not concerned about Sally being there, and he's going to show up regardless. But now the plot thickens. Sally's only been basking intermittently in the mornings and then disappearing for the rest of the day. This behaviour is common in snakes that are incubating eggs. What they'll do is they'll go out in the mornings, absorb a bunch of heat, take that back to their nest and use it to incubate the eggs, because they can't produce the heat themselves. They use the sunlight energy to warm their eggs up. For the sake of this story, we're going to assume that this is what's happening here, and Sally is about to become a new mum. This introduces some complications to the situation. If there's going to be new baby snakes around soon, we're going to land on question number 3. Is Maggie going to eat the baby snakes? So according to Maggie's owner, she's not a killer, and this is why she's been allowed outside up until now. When I hear this, my immediate response is to claim BS. I've grown up with the ingrained understanding that cats are not to be allowed outside no matter what, because no matter what their owners think, they're all killers, and it occurs to me now that I actually have no idea if this is true. So I started digging around, trying to find some research that confirmed my preconceived ideas about cat behaviour. And yeah, I know that's not how you're supposed to do it, but the thing is, I didn't find what I was looking for. There's stacks of resources out there that explain the devastating impact of stray cats and feral cats on the native wildlife in Australia, but when you narrow it down specifically to pet cats that are well cared for and fed daily, the research becomes pretty sketchy. There were several studies that looked promising that stated numbers of different species that were killed and percentages of cats that actually did the killing, but the studies suggested that there's a large subset of cats that actually don't kill. My mind was blown for about 4 seconds until I scrolled down to the methodology and learned that a lot of this research is based on data that was gathered by asking cat owners how many times they remembered their cat bringing dead things home in the past year. Thanks for trying, guys, but I'm looking for something a bit more solid than what do cat owners say they remember. There was some research that used cat-mounted video cameras, and that suggested higher predation rates than what the surveys reported, but I couldn't find much solid evidence for or against the claim that some cats kill and some cats don't. So once again, I turned to an expert for some guidance. Renee Piccolo is an honours student at Griffith University, and her research looks specifically at well-cared-for pet cats. She's gathering up poo samples from cats that have been allowed to roam free, but were also fed daily, and she's checking the samples for DNA to identify what species are actually being eaten by the cats. This is good because it doesn't rely on humans seeing what's going on, and it doesn't interfere with the cat's behaviour in order to collect the data. Now, the research isn't complete yet, but once it's finished, Renee should be able to tell us a lot more about what cats are actually killing and consuming above and beyond what their owners tell us that they remember seeing. In the meantime, Renee was kind enough to send me some of the background reading that she's gathered in preparing her research, and overall it seems pretty clear that well-cared-for pet cats are killing wildlife. What isn't clear is the extent to which it's happening and the impact that it's happening on wildlife populations. So to work out what to do, I looked at an argument by Calver et al. That was published in 2011 in Biological Conservation, and it argues that the situation warrants the application of the precautionary principle. So this mandates action to protect the environment when there is a scientifically plausible but unproven risk. So this provides a rationale for immediate intervention to protect wildlife from pet cats while we await definitive studies. So at the end of the day, we've got a lot of things to consider, and moving forward we need to make a decision about how to manage the situation of Maggie and Sally house sharing and decide what to do. There are 4 main perspectives to consider here, and you can choose your course of action depending on whether you value highest the needs of the snake, the cat, the human, or the ecosystem. And there's 4 possible courses of action that I can think of, which are 1, do nothing, 2, call the snake catcher and have the snake and her eggs removed, 3, rehome the cat, or 4, contain the cat. So I made up a Punnett square of actions versus impacts to see if I could come up with an optimal solution. It might not be the best way to communicate through an audio medium, but I'll do my best. So I've put the 4 possible actions down the left side and the 4 points of view along the top. And you can choose which action you want to consider and then cross-check that against each of the 4 perspectives and decide which one has the most positive outcomes. If you choose option number 1 and do nothing, all 4 perspectives are taking a risk. The snake risks losing her babies, the cat risks being eaten, the human risks losing her pet, and the ecosystem risks the unknown impacts of a foreign predator. If you choose option 2 and have the snake and her eggs removed, the cat and the human are both benefited, while the snake and the ecosystem are disadvantaged because removing the snake will essentially reduce the available habitat for snakes to inhabit, meaning that there'll be increased competition for resources in the spaces that are left available to them. According to a 2001 study published in Biological Conservation, over a 10-year period leading up to the study, 11,000 snakes were removed from people's homes in New South Wales alone. 20% of these were carpet snakes. On top of this, 11,000 lizards were also removed, and most of them looked a bit like snakes. That's over 2,000 removals per year, or roughly 6 per day. Now, I wasn't able to find any translocation research specific to carpet snakes, but there was a comprehensive report by Dr. Jeff Short in 2009 that looked at a whole range of Australian vertebrates, and it suggested that 28% of translocations failed due to a whole range of reasons, like being eaten at the release site. For 40% of cases, the outcome wasn't known, and for 32% of cases, the outcome was actually known to be successful. So this seems like a fairly high risk for poor Sally and her babies. If you choose option 3 and remove the cat, Sally the snake will benefit from a safer home, although she may miss out on one decent meal. I'm sure she'll be fine. But it's not a good outcome for Maggie, who loses her home, or for Maggie's owner, who loses her pet. And the impact on the ecosystem depends very much on what happens to Maggie. If she moves to a different home but she's still allowed to roam, then her impact is just shifted elsewhere. This leaves us with option 4, which is the only one that ticks all the boxes. Containing Maggie either inside the house or in a purpose-built outdoor enclosure will ensure the safety of the cat, the snake, and the rest of the wildlife. One possible downside here is that if Maggie's not happy being kept inside the house, then building a large enough outdoor enclosure to keep her happy is possibly going to be expensive. So this decision really would depend on the behaviour of the individual cat and the resources available to the owner. So given what we've learned here today, I think it's fairly clear that the best choice to benefit all involved is to confine the cat indoors. Or if you can, build an outdoor enclosure where the cat is not able to interact with the wildlife. This option also provides the added benefit of protecting Maggie from all the nebulous risks that are associated with roaming outside. Thank you to Richira and Renee for sharing your knowledge and experience, and also to Joanne, who asked the question and who cared enough about her local wildlife and her pet to want the best answer for all involved. A link to the references that I used will be in the show notes, and I'm looking forward to hearing the outcomes of Renee's research, which should be complete in the next few months. I'll keep you all updated if there's any new information that comes to light, and good luck to Maggie. I hope you find a way to live in harmony with your new housemate. Also, best of luck to Sally with your new brood of babies. I hope they all find homes where the owner is as caring and conscientious as your landlord is.
The Skeptic Magazine, the journal from Australian skeptics. Subscribe online to the world's second oldest skeptical magazine. Visit www.skeptics.com.au and click the publications link. You can also find there over 30 years of back issues free to download. The Skeptic, the magazine from Australian skeptics. And now we bring you part 3 of Speed Meet the Scientists, held at the Powerhouse Museum here in Sydney during Science Week. Folks, give me that old-fashioned archaeology. Who do we have here?
My name is James Hunter. I'm with the Australian National Maritime Museum.
And what's your area of expertise?
I straddle two things. One is maritime archaeology, and the other is naval history. So I'm the museum's naval curator.
And we're talking about the museum, which is just down the way, isn't it, in Darling Harbour?
That's correct. Yep, we're the ones down at the end of the road there. I'm just here representing the museum and talking a bit about maritime archaeology. So I'm hoping people will come along and ask me what I do, and I will tell them and hopefully not put them to sleep.
Well, that's always a good idea, I think. Now, there must be quite a very rich history of this in the Sydney area just alone, I would imagine.
That is correct, yes. There are several shipwrecks in the harbour and just outside the harbour, some of them quite significant. In fact, only about 2 weeks ago, we were doing a dive on the Dunbar, which is wrecked just outside the Heads, and it wrecked in 1857. Ooh. It was an immigrant ship that came to Sydney at the beginning of the gold rush in Australia, and it wrecked, and everyone save one person perished in the wreck. So it was one of Sydney's largest loss of life in a single maritime event.
When you say just outside the Heads, how far outside?
If you go out the Heads, hang a right to go south, and you go down probably about a kilometre. That's how close.
So very close to the shore?
Incredibly close to shore. In fact, parts of that wreck site are up against the cliff.
That's close.
Yeah.
And what was the major problem? The dangerous area it sank, or the fact that people couldn't swim?
Well, the wreck itself was a result of the fact the ship was caught in a gale. They were trying to get in through the heads, and the weather was really bad. And the captain of the ship thought that he had tacked at the right time to go through the heads, and in fact he missed it because of the conditions, and he had tacked and put the ship right into the cliffs. He basically drove the ship into the cliffs, and it broke up very quickly. High seas, ship broke up, people couldn't swim, they were wearing heavy clothing, and people drowned. The one who survived, the reason he survived was because he was up in the rigging. He'd climbed up into the rigging as the ship started to break up, and a wave basically caught him and just slammed him into the side of the cliff, and he grabbed on and managed to find a handhold, and that's what saved him. It's
like a Hollywood story, isn't it?
It is.
That's. I'm visualizing it. I'm sure all my listeners are too. That's amazing.
And the terrible thing was the guy was there for 2 days. Nobody knew he survived. He managed to kind of, when the conditions calmed down a bit, he kind of shimmied across the cliffs, and he found a spot, like a little sort of ledge that stuck out where he could sit down. And, he was there for 2 days. Nobody knew he was there. And then someone who happened to be looking over the cliff saw him and kind of heard him. He was very feeble by this point. He was dehydrated, hungry, and he was yelling for help, and somebody saw him. And so they actually were able to rescue him.
And I'm sure there are many stories. I mean, that's a dramatic one, but there must be many stories of other shipwrecks or near disasters or all sorts of things just in the Sydney area.
That's right. Many. Yes. Ships hitting cliffs, ships colliding with each other, ships catching fire, ships, whoops, I just drove it into the sandbank, that sort of thing. It runs the gamut from very dramatic, horrific loss of life to just benign sort of whoopsies.
Wow. And where can people find out more about this fascinating history?
Great place to go is the Maritime Museum. Yes, I work there, and I'm giving a pitch, but sure, we've got lots of collections related to these sorts of maritime disasters. We've got a library with lots of books and documents, and it'd be a good place to go.
And for those people visiting Sydney, it's a very conveniently placed in Darling Harbour, which is near the downtown area. But if you just Google the Maritime Museum in Sydney, you'll easily find that. Or they have a website, I presume?
They do, yes. It's anmm.gov.au.
Thank you very much.
No problem. Nice talking to you.
From one extreme to the other. Hello.
Hello.
Well, it's hardly an extreme. You're sitting right next to the guy I was just talking to. Who do we have here?
So my name's Ceridwen Boel. I'm a researcher at the University of New South Wales in Sydney.
And what brings you here to meet the scientist night? You want to meet a scientist?
I'm meeting many scientists.
Yeah.
I'm here because I'm incredibly excited about what I do, actually, and I realised, what do I have to give? I don't cure cancer. I don't necessarily, I don't necessarily invent new things, but I can share this incredible passion and this understanding of who we are and where we came from.
In other words, evolutionary biology.
Evolutionary biology, absolutely. So my background, I was really fascinated. When I was a kid, I read this book, actually not that much of a kid. I read this book, Clan of the Cave Bear, and it talked about, absolutely, and it sort of raised this idea about hybridization. So for anyone who doesn't know, interbreeding between our species and between Neanderthals or whatever other species might have been running around at the time. And it caught me. And then as I was going through my undergraduate studies, a couple of papers started coming out about DNA, hints of Neanderthal DNA in our own genomes, and it just fascinated me. So I thought, do we have any evidence for this? Do we have any fossils? Do we have any evidence of this hybridization going on? And there are a lot of fossils where they think that it may be the case, but we don't know. We didn't know what we were looking for. So I went and collected an awful lot of monkey skulls.
Where from?
All over Asia. So there's a genus known as macaques. So there are a lot of different kinds of macaque. My favourite study species was the Japanese macaque. So the snow monkey that hangs out in the sauna.
Oh, yes.
They're very cute. And it's very, very easy to tell the hybrids between them and any other species because other macaques have tails, but the snow monkeys don't. So you just look for anything with an intermediate tail and you realise that something funky is going on. So by looking at these hybridizing macaques, we were able to get a better understanding of what hybridization does to the skeleton. So what it does to the skull. The two species genomes get together, and they're mostly compatible, but there are a few little mistakes along the way. You get these little minor developmental abnormalities. So things like an extra tooth or a missing tooth or a little bone out of place in the skull. And we took all of that and applied it to humans, and we're hoping that we've got a better understanding and we may be able to identify hybrid fossils in the future.
Wow. Every time I sit down, my mind is completely blown by what I'm hearing. That's absolutely fascinating. But what are you doing right at the moment?
Okay, so I'm currently working for the Australian Centre for Biodiversity and Heritage, and we're really interested in basically everything that's happened in Australia over the last 200,000 years. So my particular project, we're actually working at some caves in Borneo, which is still relevant to Australia. So we're looking at the population, the arrival of the first humans into Australia and the spread of
migration.
Absolutely.
Right. Yes.
So we're working at this cave system called the Nia Caves in Borneo, and it's got some of the oldest human remains in Southeast Asia. At the time when they were first discovered, they were the oldest human remains in the world, and they were quite controversially linked to Tasmanian Aboriginal Australians. So we've been going back and we've been reanalysing some of the bones that were found there, and doing some new excavations, so looking for some new things. And my project particularly, I'm extracting the protein from the bones. So you may have heard of ancient DNA. It's become quite a big field, but what most people don't realise is that the protein in your bones preserves better than DNA, and it can probably tell you everything that DNA can.
I did not know that.
It's quite a new field. So DNA tends to degrade quite quickly in places like Southeast Asia, where it's quite warm and quite wet. But the protein gets bound up in the minerals in your bones, and it can stick around for a lot longer. And where DNA tells you about what you could look like, protein shows you the end result, so the interaction between your DNA and the environment. So you can see what diseases you had, what you were eating, what you looked like, who you were, whether you were male or female, how old you were, potentially. So anything we can get from DNA, we can probably get from protein, and it's going to last a lot longer in a tropical environment.
I need to take a deep breath after that. That was so interesting. And where can people find out more about you or what you do?
So our research centre has a fantastic website, actually. If you just Google Australia's Epic Story, you will find our website, and it's got information about all of the human evolution work, all of the Australian Aboriginal archaeology work, and also the biodiversity stuff. So
Australia's Epic Story.
Australia's Epic Story.
Well, thank you very much.
Thank you.
We have a real rat brain here with us, and rat brain slices, and
Well, I think I've come to the right table, folks. Not only is there champagne on the table, there's a rat brain, a human brain, and models of Homer Simpson.
That's right.
This must be a winning table. Who do we have here?
I'm Kelsey Zimmermann. I work at UNSW.
And Kathryn Baker. I also work at UNSW.
And you'll have to tell me, why do I see a rat's brain and a human brain and all these other things?
Well, we look at the rat brain to study what happens during adolescence. Adolescent humans and all the other adolescent species are kind of screwed up in how they learn and unlearn fear, and they're very susceptible to developing things like anxiety and depression. So what we do is we use rat brains like this one and rats like our little model over here to study what happens in the brain when animals learn fear and what happens when they try to unlearn it. And what we can learn from that is how the brain is different in adolescence, and hopefully from that we can find different treatments that are more effective at treating anxiety and depression in young people.
Right. So that's why and I'm holding it up here, folks. It's a little flask here with a rat. Oh, it's tiny. Rat's brain is absolutely tiny compared.
An adolescent rat brain, I believe. That's an adolescent rat brain. They're still about the same size, but not very big.
1 centimetre by 1 centimetre by 1 centimetre. Yeah, it's smaller than you would think.
But the adolescent human brain is not dissimilar in size to the adult brain?
I'd say it's probably a little bit smaller, but it's generally undergone a lot of development early in life, but actually our brain continues to develop into our early 20s.
So, actually, one of the interesting things about adolescence is that our brain is rapidly growing up until around adolescence, and then the prefrontal cortex, which is the area that's involved in a lot of decision-making and emotional regulation, starts to prune. So we lose about 50% of the synapses, which are the connections between different neurons, over the course of adolescence, so the brain becomes a lot more refined. So that part is actually, while some of the neurons are growing in different places, it's getting a lot more refined and streamlined. So that's kind of part of the reason why adolescent brains can be so sensitive to environmental stressors or other stressful situations, is because it's a very vulnerable window where there's a lot of change happening there.
Oh, I remember it well.
Yeah, don't we all?
We certainly do, yes.
Yeah, so another part of research we have in our lab is actually looking at the effect of junk food diets in adolescents. And we find that actually adolescents are much more sensitive to these type of diets than adults are. So in some of my work, we've shown that an adolescent rat is impaired in spatial memories, so earlier than, say, an adult rat is, fed the same diet.
That's where Homer Simpson comes in.
That's where the Homer Simpson is. So I've got a diagram here about how we test for spatial memories in the rat. So we've got two Homer Simpsons here, and basically rats like novel things. So I've got a little diagram, a little toy rat, and he's like sniffing both of them. But after 5 minutes, when he's back in his home cage, I'll move one of these Homer Simpsons, and he has to go back into the cage and find which one is the novel one. And so a normal rat will spend more time investigating and sniffing around this Homer Simpson in the new location, but the rat that's been fed this high-fat, high-sugar diet will forget, and it'll like go and explore like both of them equally. So that's how we test spatial memories in rats.
Wow, I have come to the right table. Where can people find out more about your research?
We have a website. Our boss is Professor Rick Richardson at UNSW, and so we're under the Richardson Lab, and there's a lot more pictures and information about the different research that we do.
Thank you very much.
Thank you very much.
Thank you.
And it's time for another table. Hello.
Hello.
And who do we have here, and what do you do?
My name is Titia Benders. I'm a developmental linguist, which means that I study language as a phenomenon and especially how people learn it. So how it develops from young people just before they're born until about 5 years of age.
Wow. And from what my very limited understanding is, that is exactly the time where the human brain is like a sponge, listening and learning at an incredible rate.
Yes. And that sponge is present, that sponge is already there before birth. And the ears are also functioning before birth, so the sponge starts absorbing before birth. I think we always have to keep in mind that birth is just a benchmark event in life, but it's not when development starts. Language development starts slightly before that, and especially the first and second year of life are incredibly important in building the foundation for further language development. And I mean, children get jokes only when they're maybe 8 or 9 or 10. They're still developing the subtleties of language, but the big foundation blocks are created within the first 2 years.
Yes, you're right. I think there are many things that children will grow to understand. But if you're talking about jokes, I mean, being an uncle myself, even when the kids were 3 and 4 and 5, they found things so funny, and you can make them laugh so hard. Isn't that interesting that the sense of humour kicks in so early?
I find it very hard to speak to that because I'm a phonologist. So what I can observe is anecdotally how children around me develop language and develop jokes and develop a sense of humor, and sometimes can find something very funny because they recognize that everyone around them is laughing, so they're laughing at the same time because that seems to be the thing that we do. But they might be looking at context rather than actually understanding why it's funny.
Yes, you make a good point. So we think about something like a pun, for example, which needs some complexity of thought to understand and appreciate the humour. That really wouldn't come in until a bit later, you say?
Until a bit later, and I don't want to be quoted really putting an age to that. But what we can observe is that language competence is knowing the language, but it's also knowing how to use it in a social context. And if you know to laugh at the right moment, you may appear like a competent language user, but you may not have gotten the joke.
That's interesting. That's fascinating. And again, folks, it's one of the many fascinating scientists here tonight. Are you online? Where can people find out more about what you do?
We are the Child Language Lab at Macquarie University, which means that we are also always looking for little scientists to help us with our research. I think the best thing to do would actually be Googling for Child Language Lab Macquarie University and then find us.
There's the clue, folks, if you're interested in that sort of thing. Well, thank you very much.
Thank you very much, and have a good night.
I've come out here to the courtyard in front of the Powerhouse Museum. Hello, who do we have here?
Jason Drury.
And next to your telescope, and you're just telling people about Jupiter. And what are you finding is fascinating people tonight? Is it because you're focused on Jupiter?
So part of it is Jupiter and the fact that you can actually see this detail on the surface. So you'll see some stripes, which are the different gases that make up Jupiter. And you're actually seeing detail on the surface of a planet that's 720 million kilometres away. It's pretty mind-blowing stuff.
How many light seconds? There's one for you.
It's actually about 45 light minutes, so times that by 60.
Minutes, right. Well, you got me. Well done. Good answer. 45 light minutes.
About that, yes.
That's impressive. And let's gaze up. I'm gazing up now.
Yeah.
There it is, that one.
Yes, so that is it.
That is it. You get your laser out. Yes,
this one right
that's
here,
right.
This is Jupiter right here.
That's Jupiter. And people are lined up to look through your telescope. And this bright one down here.
So that one right down near the horizon towards the west is actually Venus.
That's Venus. Okay.
So we've got Venus over here.
Yeah.
Higher up we do have Jupiter that we just saw. A little bit further along in that line, that's Mars. Okay, we've got Saturn just over here. And a little bit further down we've got Mars just over here.
And doesn't Mars look bright tonight?
It does. It's actually almost, it's just gone past the closest point to Earth for the last 3 years or so. And it was just at opposition. So that is where the Sun, Earth and Mars all line up in one nice big straight long line.
Well, let's wander over to your telescope now, folks. I'll just bend over here and have a look through this little eyepiece. And wow, look at that. And you can see 4 of the moons.
Yes, so 2 above and 2 below.
Actually, I can see 5.
So I'm not 100% sure if that 5th one is actually a moon. I'd have to double check.
It could be a little star.
It could be a star way, way, way back behind it all. But we do have the 4 largest moons of Jupiter. We've got Io, Europa, Ganymede and Callisto. So they're the 4 large ones that we can see that almost look like stars.
And they're right there. And you know what? If I look up with my naked eye now, and I'm looking towards Jupiter, am I fooling myself to think I can see one of the moons above it?
So whilst it may look like a moon, it's actually a star. That one, I can never pronounce it correctly. It's pronounced something along the lines of Zimmermann Canabi. And it's actually a binary star. So it's 2 stars orbiting one another and spinning around in space.
All right. Well, thank you very much for that little lesson. And as I can see, all the people lined up here, it looks like a very successful night for astronomy.
Indeed it has been. And for Sydney Observatory as well.
That was the final part of our 3-part series of Speed Meet the Scientists. A big thank you to Frankie Lee from the Powerhouse Museum for making it all possible. Thank you for listening to the Skeptic Zone this week, and a big thank you to Angus Firth, who was my intern for this episode, who came along and edited and mixed a lot of today's episode. And he did a fantastic job. Well done, Angus. I think you'll be doing some more Skeptic Zone editing in the near future. Now, don't forget, of course, the big Skeptic Con convention coming up in October. And if you're in Sydney, please come along to the free event on the eve of Skepticon, which is a big welcome event at the Occidental Hotel in Wynyard, where we normally have skeptics in the pub. Link on this week's show notes. If you want to come along, please RSVP. Remember, it's free. It's just nice to know the numbers. We're going to have a big trivia night as well. We're going to take up 2 floors of the Occidental Hotel. It should be a really great night. Of course, while you're there, if you haven't done so already, please book your tickets for Skepticon. Coming up on next week's show, we're going to be chatting to the challenge coordinator and investigator from Australian Skeptics, Ian Bryce. We've had to bump his segment from this week to next week because this week's show was quite full. Also, we chat to Celestia Ward from the Squaring the Strange podcast, a wonderful skeptical podcast coming out of, well, I guess it's coming out of Albuquerque, New Mexico and Las Vegas, Nevada, with her co-hosts Ben and Pasquale. Anyway, we're going to be chatting to Celestia Ward, and she is, if you might remember, some people might know, she is a fantastic artist. In fact, the caricature of me and Maynard we both use on our business cards was done by Celestia. A big thank you to those people who continue to support the Skeptic Zone with donations and PayPal micropayments and via Patreon. And new people, and you know who you are the only reason really the show keeps going, because I certainly couldn't afford to do this show, the time it takes and the equipment and all the rest of the things, without real financial support from listeners like you. And if you've never thought about contributing, maybe even $5 a month goes a long way here at the Skeptic Zone via Patreon or whatever. Links at skepticzone.tv. Much appreciated by myself and the whole team here at the Skeptic Zone. And a big shout out to my friends enjoying Dragon Con in Atlanta, Georgia right now. I've been following various threads and postings and videos on YouTube and Facebook and Twitter and all sorts of things, and I'm so jealous. I wish I was there. I hope maybe I can be there next year. I haven't been to Dragon Con for about 8 years. My first Dragon Con was 10 years ago, and that's in fact where I recorded the first segment for the Skeptic Zone, which I guess in a way this weekend marks the 10th anniversary of the first bit of recording for the Skeptic Zone, so there you go. But for this week, this is Richard Saunders signing off from Sydney, Australia. You've been listening to the Skeptic Zone podcast. Please visit our website at www.skepticzone.tv for show notes, contacts, and to access the back catalogue of episodes going back to 2008. You can follow the Skeptic Zone podcast on Twitter at Skeptic Zone, visit our Facebook page, or leave a review on iTunes. You can also support the Skeptic Zone via Patreon or PayPal. The Skeptic Zone podcast is an independent production. The views and opinions expressed on the Skeptic Zone are not necessarily those of Australian Skeptics or any other skeptical organisation. Hello, all the people who listen after the music, the post-music gang, or maybe somebody can come up with a name. I've had more requests for the dice game, so here we go. I've got a die in my hand. It's a 10-sided die. I'm going to roll it 3 times, which will give me a number between 0 and 1,000. So you use your predicting power now, folks. Predict away. Before I roll the first number, have you got your number between 0 or from 1 to 1,000? Good. I'm just waiting for you. Yes, you've got it? All right, here we go. Shake the die. 100 and 5. 105. Did you get that? For now, ich sage Tschüss.